Hi, this is Sarah Meixel with the Pig Site, working in collaboration with Lifestart Swine. And today we're online with Dr. Marcelo Gottschalk. He is a full professor at the Department of Pathology and Microbiology of the Faculty of Veterinary Medicine at the University of Montreal. Dr. Gottschalk is the director of the International Reference Lab for Swine Pleuropneumonia and the International Reference Laboratory for Streptococcus suis, which is an important swine and human pathogen. He's also a consultant for the World Health Organization on human disease caused by strep suis. Thanks for being with us today, Dr. Gottschalk.
Thank you very much for inviting me to this interview. Thank you.
Sounds good. It sounds like you have a busy life and you are on the go with lots of activities related to swine and human health, actually, is what it sounds like, right?
Yeah, that makes me travel a lot. So I am very often outside the country, but it's okay because there are very nice experiences, especially with Southeast countries where the human disease is extremely important, such as Vietnam, Thailand. So it's we have very nice collaborations with that.
Very good. In all of this, and we'll dive into some of your research and stuff, it does remind us with diseases like this connection between human diseases and animal diseases, that zoonotic connection that we have with especially some of these diseases that you work on.
Yeah, that's true. And there are two connections there. I mean, the connection of human transmission, I mean, particular zoonosis, which can be transmitted from animals to people, but there is also the question of antibiotic resistance, and strep suis, which has been treated with a lot of antibiotics in the past. We have a lot of resistance in other bacteria that are acquiring that resistance from strep suis. So it's we have a double problem of, public health from the zoonotic point of view, but also from the antibiotic resistant point of view.
Interesting. Well, we'll dive right into our questions that we wanted to talk about today. Tell us about your work and research to develop strep suis vaccines.
There are practically no commercial vaccines against strep suis. So that is one of the most important problems because it has been treated and prevented using antibiotics for many, many years, which is more and more difficult, almost impossible in Europe. And so there is a need for vaccine. The problem is, so far, as I mentioned, there are no commercial vaccines. So people have been using autogenous vaccine. That means the strain isolated in the farm is used to produce a vaccine, which is a kill vaccine because bacteria are killed. The problem is laboratories producing this type of vaccine. Each lab will produce the vaccine in a different manner. There are no standardized protocols, and each company will keep the secret of the production of this autogenous vaccine. So an autogenous vaccine produced in one lab and another produced in another lab are completely different products. So on one side, we have been studying the production and optimization for the autogenous vaccine, how to produce an autogenous vaccine, the use of adjuvant bacterial concentration, the use of different strains or different bacterial species in the same autogenous vaccine, as most of the practitioners will use. So we have been working on how to optimize this autogenous vaccine. We are not an autogenous vaccine company because we are academia, but we can give some tips to those companies trying to improve the production of autogenous vaccine. However, the future is not necessarily autogenous vaccine. It's a more universal vaccine, commercial vaccine, because these are produced under very strong regulation. They are tested for protection. Autogenous vaccines are never tested for protection. So we are in parallel developing some candidate for vaccine production. One of the candidates, this is done with my colleague, Mariela Segura. She is our immunologist specialized on strep suis. She's also my wife, so it's very easy to work together on the on the
Very
subject.
good.
She's a very smart woman. And what we are developing is a synthetic vaccine, which means the bacteria has a capsule. The capsule is a kind of shield that protects bacteria, and it is composed by sugars. And sugars are not very immunogenic. There are not a lot of antibodies against the sugar that happen in humans, in all species. But if you get antibodies against the sugar, they're very protective. So in humans, they will use the complete capsule to produce a vaccine. The problem is that it's extremely expensive. You can do that in humans, but you cannot do that in pigs. So we have synthesized a little part of the capsule, completely synthetic. This is linked with another protein, so the protein will make the sugar visible for the immune system, and the immune system will produce antibodies against this little thing, which is synthetic, which will recognize the capsule and will protect. We have already tested at least twice. We look for different little parts of the capsule. We found that one of that part is protected. And we are developing now that kind of vaccine in collaboration with the industry. So this is one of our main topic of research now, developing a synthetic vaccine against strep suis. There are only one vaccine in humans using this kind of technology. There are no vaccine in veterinary medicine, not only in swine, but in veterinary medicine. That will be probably the first vaccine of this type.
Interesting. Why are researchers struggling so much to develop a successful vaccine against strep suis?
Two different things. One of the problems is to have a very good vaccine candidate, because strep suis is a very low immunogenic bacteria. What does it mean? It means that strep suis does not induce a lot of antibodies in the pigs. So you need to find a good antigen that will induce antibodies, and if possible, to protect against different strains. So the problem is strep suis is heterogeneous. There are different strains, and you need to have antigen that will cover all those strains. This is one of the main problems. But the second is, let's say that you have the best vaccine candidate, protecting 100%. Excellent. The problem is, who are you going to vaccinate? So you say, okay, to reduce cost, we would like to vaccinate sows.
Okay.
Good. So you vaccinate sows. The problem is strep suis will induce disease in post-weaning piglets from 4, 5 weeks of age until 9, 10 weeks of age, just before going to the growing finisher phases. So the problem is, if you vaccinate sow, you need to have enough maternal antibodies to cover all the post-weaning period, which is very difficult. Why is difficult? Because colostrum intake is not homogeneous. It's very heterogeneous. We have many studies where we demonstrate that even siblings, I mean, from the same sow, they will have, one of them will have very high level of maternal antibodies, the other one very low, because it depends on the access to have colostrum. So the problem is that sows are vaccinated. In theory, all piglets will have a lot of maternal antibody, which is not true. You will have half of the piglets with very high antibody level, the other piglets with low antibody level. So it's very difficult to make these levels last until the end of the post-weaning. So it's difficult to have that protection. So which will be the alternative? The alternative is to vaccinate piglets. Okay, every is most costly, but let's say that we can vaccinate piglets. When are you going to vaccinate? If you vaccinate piglets, I don't know, 3 at weaning and 5 weeks of age, you need always 2 doses because Strep suis is low immunogenic. Animals will be protected too late. I mean, at the end of the post-weaning. So it would be better to vaccinate. This is done in USA in some farms to vaccinate, at 1 week of age during processing. You give iron and then you give the vaccine at the same time, and a second dose at 3 weeks of age at the weaning. One of the problems, we are studying that. One of the problems is when you have maternal antibodies and you give a vaccine, many times there is an interference because the antibody from the mother will neutralize the vaccine that you are giving to the piglet.
Right.
So vaccinating very young piglet may be dangerous also. So we are studying now the degree of interference with maternal antibodies. So the problem is, vaccinating very young animals to be protected in the post-weaning, you may have problems with interference. Vaccinated sows, you may have the problem not covering the whole post-weaning period. So this is one of the causes why we still do not have a very, very good vaccine, because even with a good vaccine, it is difficult to decide to whom we should and when we should vaccinate.
Right. And is strep suis one of those pathogens that kind of is evolving and changing? Is that a challenge with this, or is it stay pretty steady and it's not, you don't have a new strain every year, every 3 to 5 years?
It's a very good question. There are some new strains that are emerging, but usually they are, they will not evolve very, very fast.
So it's pretty consistent, right?
Yes, but another problem that we have that the strains present in Europe are completely different from the strain present in North America.
Oh,
very different.
I guess that's not like with PRRS, right? Too. I mean,
yeah, it happened with PRRS. It happened with that, swine pneumonia. It will be the same. They are not the same serotype. And that is another problem for the vaccine, trying to cover everybody because the strains are very different. As I mentioned, we may have sometimes, 15 years ago, we have the emergence of a very highly virulent strain in China, which caused a lot of people dying because it was very high virulent.
Yeah.
And this is a strain present only in China. So sometimes you may have that kind of emerging strains, but usually there is not a lot of change.
Got it. Okay. Well, I have a lot more questions about that, but I will, we'll keep moving on with our questions. What role do co-infections play with strep suis?
Just to explain to you, when we talk about co-infections of strep suis, if you go in the literature, you will find that almost every pathogen will collaborate with the strep suis to cause disease, which is usually not true. You have one main, and I guess you know what I'm talking about, one main pathogen, which is PRRS, obviously, and then which is the relationship. It is extremely clear when you have a herd which is unstable, you will have problems with the strep suis. Which is curious that we do not know how this works. In what I mean by that is this collaboration with the virus, with the strep suis, obviously affecting the immune system of the bees. This is very general, but exactly how they are working together, nobody knows. But this is what everybody knows, that we have a herd which is really not stable or with a high virulent strain. You have a country like Spain, they have a very high virulent strain of PRRS. And they have a lot of problem with the strep suis in the last year because, the number of cases increased a lot because you have really a predisposing factor. Influenza is another predisposing factor, which is also important for the strep suis. The other PCV2, mycoplasma. I mean, there are no studies. I mean, we have tested co-infection with mycoplasma. We didn't find anything. With Lacerola parasuis, we didn't find anything because as everybody was talking about, there is a connection between those pathogens. We test that, but we couldn't find any association. So to answer your question, peers in the first place, influenza in the second place, there is Olszewski, but I mean, I'm talking in countries where, you have Saudi Arabia, which is US and Canada, we are free of that. But in those countries, still people, I never worked with that, but people said that maybe some connection, but others are less important than those I mentioned.
What about general health and post-weaning diarrhea? Are these associated to risk for strep suis cases?
I believe this is the most interesting question that you have asked. And because, for the strep suis, everybody has, I'm going back a little bit, but everybody has always described the infection through the respiratory route. We do not know how strep suis go to the bloodstream to cause systemic disease, and we have demonstrated that if we infect animals in one room, there's no direct contact, but you have common air, and 1 week later, the other animals will develop disease. So we know that bacteria can be transmitted airborne. 5, 6 years ago, a second hypothesis came to the scientific community proposing that the strep suis can infect through the intestine. In human, in Thailand, Vietnam, Laos, they will get sick by ingesting raw pork meat or blood in some dishes that they use. That you see the dishes and looks like ketchup, but it's not ketchup, it's blood, and it's fresh blood from pigs. So that's a common way of infection in human. So it's why not in pigs? So there are a lot of research, and nobody could demonstrate so far that the intestine is the way how strep suis can reach the bloodstream. But if you go to the field, you will see that the first piglets affected by strep suis are the strongest, the nicest piglets. You are not expecting that. You expect the opposite. The weakest piglet will be more susceptible to the disease. It's not the case usually. What we believe is that the biggest, the nicest piglets are those consuming a lot of milk from their mother, and are those who will have more trouble to adapt to solid feed. So in the transition, these animals
feed, right? I get it.
That's right. And during 2-3 days, these animals are almost not eating at all. And all of a sudden, they become, more and more adapted to the feed, and then eat, and they try to recover, and they eat too much. So the curve is like this, going down and then going up. So the intestine is completely affected. That means that the microbiota of the intestine, the inflammation level of the intestine, everything changed, and this became a predisposing factor. So the health of the intestine seems to be extremely important for developing the disease. Not necessarily because bacteria is going from there to the bloodstream, but because the animal is really not healthy. There are a couple of studies. I don't know if it's reproducible, but at least 2 studies, different studies, show that vaccination using a live vaccine against E. coli reduced the cases of strep suis. in the
Mhm.
herd. I don't know if it's a direct connection, but what I know, if you have more diarrhea, if you have, in Europe, they cannot use zinc anymore, so they have more cases of diarrhea. Having more cases of diarrhea, you have more cases of strep suis. So it seems to be a connection there, and it's probably due to homeostasis of the intestine, which is affected. That is very interesting question because people are looking at that more and more because in the past people said that no relationship you have. There is no relation between diarrhea and meningitis. I mean, how you can? Yes, you can.
It is interesting because it goes back to what we started talking about is the things that you're learning from human health can be carried over to animal health, right?
Very different, very different because I mean, people thinking about the intestine as a root of infection, that makes sense because in
Right,
humans, as I mentioned, China is different because in China, people will eat cooked pork. So it's
right.
not the problem is the direct contact, with hands with sick animals. But in the rest of Asia, in Southeast Asia, 99% of cases are after ingestion of contaminated food.
Mhm.
So bacteria in humans go through the intestine. It does not seem to be the case for pigs.
Yeah,
So probably related to uh, the type of the feeding, which is different, I mean, this is completely different in humans and in
right.
pigs, but nobody knows exactly why, but it is the case.
Interesting. Very interesting. Well, with your experience across pig producer countries, what are the most effective contingency strategies or the most effective system to reduce the risk of strep suis outbreaks?
Um, okay, perhaps this is a politically incorrect answer, which is you would like to have the most effective method to control strep
Mhm.
suis with antibiotics,
Right.
which we have been doing for many, many years. The problem is that we cannot do that anymore. We are trying to reduce that more and more. So, but that worked very well. And that is why 15 years ago, strep suis was considered, as a second invader, which nobody cares a lot. And
Right.
today is the most important cause of bacterial disease in post-winning piglets around
Oh,
the world.
right.
So it's the most important one. But it's not necessarily because there are more virulent strains that we discussed earlier. It's mostly because they cannot use antibiotics. So if you ask me in the first place how to control, which is the most effective way to control, first to control Pierce infection.
Mhm.
And obviously, there are many farms which are completely free of Pierce influenza and they are affected by the strep suis. That can happen because there are very virulent strains of strep suis, but this will help a lot. And reducing stress, reducing environment. Strep suis is a disease that sometimes if you have a very high virulent strain, you will have disease whatever you do.
Mhm.
That,
Right.
but in North America, I will give you because one of my laboratories is a diagnostic lab at university, and obviously we are specialized on strep suis. And I talk very often with the practitioner and taking a look for the last 5, 6 reports, we see that there are 5, 6 different strains, different serotypes of strep suis
Huh.
causing disease. Because they usually contact me to say, I would like to choose a strain for an autogenous vaccine.
Right.
Looking at those results, what I usually tell them is strep suis is not the origin of the problem, is the consequence of the problem. You have other problems in the tonsil of the pigs. There are a
Mhm.
lot of strep suis. They are common inhabitants. So depending on the condition, sometimes one strain will invade, let's say the overcrowding, difference of temperature between the night and the day. There are many, management or
Right.
environmental factors that will induce the animal to develop disease. But if you try to attack each time that a strain of Strep suis appear, it will not work. You need to attack other things to control because you don't have a very virulent strain. You have strains that are opportunistic and invade in the moment where the conditions are the right conditions. So I would say that if you have a very virulent strain, so far, there are not a lot of options. You may try a good autogenous vaccine and try to see if this may help. And with virulent or less virulent strain to control all other factors that will increase stress from the animals. We talk about diarrhea, for example, or all other factors that will induce stress on the animal. Controlling those factors, it will probably help to control the infection. But it's still one of the most difficult diseases to be controlled today.
It's a lot to take in. You know what I mean? It's so complex, right? And the co-infection
Absolutely.
adds to that, right? The co-infection part of this all adds in. So
Absolutely. Absolutely.
very good. And our last question here is a little bit kind of forward thinking. We're going into an era of disease-resistant pigs. Is that an option for Strep suis? Do you see that as a potentially a gene editing opportunity or otherwise?
Nothing that is known today, but talking about the future, nothing is impossible. But we do not know which are the receptor, because as I mentioned, Strep suis is a normal inhabitant of the upper respiratory tract. That means you go to the tonsil of any pig in the world, and you will isolate Strep suis. They are present normally. So the question will be, because these bacteria are colonizing the tonsil, part of the normal flora. So you would need a pig with modification in the genes that will become resistant to disease, but not to infection, because this is the other thing when we talk about the vaccine, that we cover all the strains of Strep suis. See, that would be ideal to have a vaccine to protect. Yes, but are these vaccines modifying the normal? It's a thing that you need to think about. The normal Strep suis in tonsil, you don't want to reduce that a lot because they are normal flora. You don't know how the animal will react if you. It happened the same with E. coli. I mean, now we're thinking to eradicate E. coli from the intestine. You want to eradicate virulent strains of E. coli, but E. coli is a normal inhabitant of the intestine. Strep suis is a normal inhabitant of the tonsil. So nothing is impossible, but I think that it will be extremely difficult to have genes modification in the host that will, the animal will become resistant to develop disease, but not resistant to be colonized by the Strep suis. I mean, nothing is impossible, but I believe it will be very difficult, very different with peers, because you won't animals free of virus.
Right.
So that's okay. I mean, you can target, as it has been done, you can target genes because, if the animal cannot be infected by the virus, that's better. It will be different with the Strep suis. So it's not so easy, a little bit more complicated.
Right. But we never say never, right? In this industry.
Never. I have been working on this for more than 30 years. And, I made mistakes on when you think these things that shouldn't be like that, and you test it, it is like that. So never say never, as
because breakthroughs do happen, right? And we don't know exactly what technology we're going to have available to us in 10 years, right? So
we should be very humble because biology is biology. The technology is changing extremely fast.
Right.
And you never know. So it's we can say what we think, that but perhaps it's the opposite, and it's
Yeah.
okay. That.
Yes, you never know. We might be watching this video in 10 or 15 years and be like,
Oh, listen what he
right?
said. Yeah.
How could
Yeah,
we have said that, right? Like things like that happen, it feels like. I don't know. I feel that way about my cell phone, right? Like I never thought it would do all the things that it does, but
Uh, I will tell you what very, very fast.
yeah,
When in the year 2000, there was this outbreak in China, killing a
right,
lot of people. I began to receive phone calls in the middle of the night because there are journalists in Asia,
right,
and they were asking me, what do you think? There is a strep suis killing a lot of people, and my first reaction was, this should be other thing. Cannot be strep suis killing so many people all of a sudden, and it
right.
was a strep suis. But my first reaction was, this is strange. And a lot of people in here at the university made fun of me because that reaction that I had at that moment was in a different journal. They said, "Well, Dr.
Right.
Gottschalk think that perhaps should be other thing than a strep suis." It was not, it was a strep suis. But that's the case when you say sometimes, okay, things are different because
Yeah.
25 years ago, you said strep suis will emerge and kill a lot of people. So what? I mean, it's
You'd be like,
a
no
swine
way, right?
pathogen. Swine pathogen. What it will? It's not a virus that will, jump but it was the case. So yes, as you said, that
We
can
learn as
happen.
we go, right? Knowledge is power.
Absolutely,
We just learn as we
absolutely,
go. So very good. Well, hey,
absolutely.
thank you, Dr. Gottschalk, so much for all the information today. This was really enlightening. I learned a lot. So thanks so much for being with us.
It was really my pleasure. Thank you for the interview.
We do appreciate it. This is Sarah Meisel with the Pig Site, working in collaboration with LifeStart Swine.